So this video summarizes a project that was looking at the relationship between orbit characteristics and species ecology in macropods. The family Macropodidae has extreme variation. A lot of people just think of it as kangaroos and wallabies, but there's many more species and like genuses from like tree kangaroos and rat kangaroos. The ecomorphology of the eye in these species is very understudied. It's been studied in mostly primates, but there is still a lot to be explored within Macropodidae. You can look into things like visual acuity, binocular vision, and field of view, and link that to a species ecology. Working with some other groups that were looking into different groups of marsupials, we looked into our pilot study where we started to experiment with different cameras that we could take our measurements with, and eventually realized that it's very important to keep consistent camera settings and positioning of the orbital features in relation to the camera. Eventually we decided to use a camera system that would not have as much human error involved, which was this Canon EOS 5 DSR camera. It also became particularly important for us to keep the lighting consistent, the distance from the camera, and this was all monitored by Capture One Pro and Zareen Stacker software. Next up, starting with my smallest specimen, I actually started taking the photos I was going to use to measure the angles. So I took the dorsal photos here, keeping all the settings same and aligning the skull to match the blue calibration lines. After each image was created and had a scale bar and its label, it was then transferred over to measure the angles by marking out the landmarks, which were there and there along the plane across the middle. Then an online protractor was used to record the convergence angle. So now I've started to place the lateral view of each skull. So I'm doing a red kangaroo right now. I'm using foam blocks to position it so it is a perfect lateral view. I'm lining up the crosshairs from the top and the bottom, the same as I did for the plan views. And also, I need to make sure the tooth line is a perfect vertical line, and I have the lights and camera set to the same settings as I had before. So as each skull was a different size, I had to adjust the distance from the camera each time by like moving that bit up and down. And like this was the only thing I could change to then be able to refocus it and not have any inconsistencies in the photos I've taken that will change the angles. And each time it was still aligned in those two blue lines there, which were like calibrated to keep consistent size of an image. A scale bar has also been put into every picture that was taken, and also kept at like the same height as the orbit, so then there would be an accurate representation of measurements. And by using this ID card for each individual, you could actually see if there was any variation from maybe age, sex, or any notable features that were recorded on this little ID card, which is useful in analysing your results. When it came to actually recording the angles, we found it hard to find the landmarks for some of them. So we noticed in an extinct species, one of our landmarks, the post-orbital process, was directly above the process just below. So we use this as an analog to find our landmark when the post-orbital process wasn't very prominent. So I have now started taking the measurements to estimate the body mass of each individual. I'm taking measurements of the length by measuring its longest points from its front teeth to the back, and then for the width, I'm taking measurements of its widest point from each side. So for the manual measurements, I was mostly just using calipers and a ruler. And these measurements gave me estimations of body size and orbital size in like the area of the orbit. I took these measurements for each species I was looking at, which took a very long time. I was looking at 5 different groups, all with varying niches, starting with the largest red kangaroos, then wallaroos, tree kangaroos, wallabies, and rat kangaroos. So after compiling all of this data and carrying out some statistical tests, I managed to make some figures that could actually answer the questions that I came up with and disprove or prove my hypotheses. I did find a statistically significant relationship for one of my questions, which was whether there was a correlation between relative eye size and body size, and there was a negative relationship which shows that the smaller a macropod was, the more likely it was to have larger eyes in comparison to its body size. This was particularly noticeable in the wallabies, which were Petrogale lateralis and Petrogale xanthopus, the yellow-footed rock wallaby and the black-flanked rock wallaby. They both have particularly high eye size in relation to body size, which was possibly associated to predation. Another one of my questions was based on the convergence of the eyes and the angle that represented their binocular vision. And there was noticeably higher convergence in the tree kangaroos and the rat kangaroos. The tree kangaroos particularly having a high convergence angle at around 58 degrees. And this was thought to be associated with their habitat, as they spend a lot of time in like the rainforest with lots of canopy cover, where they need better eyesight to be able to see because it's darker, and also for moving through trees they need better depth perception, which is correlated to the convergence angle. And for the rat kangaroos, which also spend time under the undergrowth and in shrubbery and in burrows, they need to have a better eyesight and a larger convergence angle, which can help them move very quickly to escape predators, and again spending a lot of time in their habitats, which was a rainforest as well. My last question was asking about the vertical angle of frontation, so measuring the angle of kangaroo eyesight from its tooth line up and seeing how far up they could be measured. And this graph shown here does not actually show much variation or significant results. This could either be a methodology issue, or it could be that there's no variation in the species that we looked at. So looking at more species with more specimens of each species can help answer these questions fully. But overall, there was lots of variation in the macropods, and further study widening to maybe marsupials in general could show even more variation that can be associated to diet, locomotion, habitat, and lifestyle.